Hello, this is a video lesson on radiation biology. There are several parts in this series. To begin with, we're going to start with part 16A on human biology. The effect of X-rays on humans is the result of ionization at the atomic level. The result of ionization is absorption, and therefore we're going to have damage. Early effects of radiation are those that occur within minutes or hours after radiation exposure, and late effects of radiation exposure are those that are not observed for months or years. It can take as many as 20 years for many of the late effects to transpire. Radiation biology is the study of the effects of ionizing radiation on biologic tissue, and keep in mind that even small doses of X-rays are harmful. So looking at the atomic composition of the body, we noted that 60% of the body is comprised of hydrogen, 25.7% oxygen, next is carbon, then nitrogen, and so forth, down to trace elements at 0.8%. As far as the molecular composition of the body, 80% of the body is comprised of water, 15% is comprised of protein, 2% lipids, 1% carbohydrates, 1% nucleic acids, which includes both DNA and RNA, and then 1% other. In terms of molecules, we have organic molecules and inorganic molecules. Organic molecules are macromolecules, and that means that they're very large, sometimes comprising of hundreds of thousands of atoms. There are 4 types of organic molecules, and they all contain carbon. Organic molecules contain carbon. That includes proteins, carbohydrates, lipids, and nucleic acids. Inorganic molecules include water and mineral salts. So let's begin with proteins. Proteins are long-chain macromolecules that consist of a linear sequence of 22 amino acids connected by peptide bonds. Those amino acids are made up of SNOT, which is carbon, hydrogen, nitrogen, oxygen, and trace elements. The functions are that provide structure and support. They function as building blocks, and they function as enzymes, hormones, and antibodies. Lipids and carbohydrates are both comprised of carbon, hydrogen, and oxygen, but their composition is different in that the number of carbon, hydrogen, the ratio of carbon to hydrogen and oxygen differs. Carbohydrates are just watered carbons in that they are, for every 1 carbon and for every 1 oxygen, there's 2 hydrogens. So the original name of carbohydrates were watered carbons, and then they changed the term to carbohydrates. Carbohydrates provide fuel for cell metabolism. They're short-term energy stores for the body. They are structure for cells and tissues to provide shape and stability. There are 3 types of saccharides. One is called a monosaccharide, and mono means 1, so it's 1 sugar. Then disaccharide is the sugar that has 2 units of a simple sugar link. Polysaccharides are many molecules of a simple sugar. Lipids are water-insoluble organic macromolecules. They're composed of 1 molecule of glycerine and 3 molecules of fatty acid. Glycerol is a syrupy, sweet, colorless, or yellowish liquid, and fatty acids are constituents of amino acids. The functions of lipids include structural parts of cell membranes that support and protect organs. They're fuel, which is a reservoir for long-term energy store for the body. They're concentrated under the skin and serve as a thermal insulator from the environment. They provide essential fatty acids needed for growth and development, and they assist in the digestive process. Nucleic acids are large, complex macromolecules made up of nucleotides. And a nucleotide is a base sugar phosphate combination. We have 2 types of nucleic acids. As you can see here, we have DNA and RNA. DNA is located in the nucleus only. It contains all hereditary information of a cell, and it's the command or control molecule for cell function. Therefore, that makes it a radiosensitive target molecule. It controls cell function and it contains hereditary information if it's a germ cell. DNA has a double helix configuration. That means it has a backbone or rails, which are alternating deoxyribose and sugar, which are along the side rails of the molecule. The rungs of the ladder are going to be the nitrogenous bases joined together by hydrogen bonds that join the bases to the side rails. We have two categories of bases. We have purines, which are adenine and guanine, and we have pyrimidines, which are cytosine and guanine. As far as base bonding, if you look at the rungs of the ladder, they're bonded so that they have A and T, which is adenine and thymine, on one rung, or thymine and adenine. Alternatively, they could have cytosine and guanine, or in order of guanine then cytosine. So there's four possible ways for bases to bond on the rungs of a ladder. The order of the base pairs is going to make up the genetic traits of the individual or produce different cells with different functions. RNA is found in the nucleolus, the nucleus, and the cytoplasm. It's formed from DNA but carries out its function in the cytoplasm. There are two types of RNA. There's messenger RNA and transfer RNA. RNA is strategically similar to DNA except it has a single spiral. Uracil replaces thymine and it has ribose sugar versus deoxyribose. Water is the most abundant and simplest molecule in the body. It's inorganic, by the way, which means it has no carbon. It provides some form and shape. It assists in maintaining body temperature. It enters into some biochemical reactions. Inside the cell, it's a solvent by dissolving chemical substances, acids, bases, and salts. Outside the cell, it's a transport vehicle for materials the cell uses or eliminates. Mineral salts are ions within the cells, and these are also called electrolytes. Their functions are essential for proper metabolism. They maintain proper proportion of water in the cell, which is called osmosis. It's necessary for proper cell function. They create energy and they conduct impulses along nerves. And these also are inorganic or non-carbon based molecules. Now let's look at the cell structure. There are two main structures which include the cytoplasm and the nucleus. The cytoplasm contains all cellular components except DNA, and it's the location for all metabolic functions of the cell. And then there's the nucleus. The nucleus is a round mass of protoplasm that contains one or more nucleoli and chromosomes. It contains DNA, which is the only location that DNA is present in the cell, RNA, protein, and water. Its function is it controls cell division and biochemical reactions within the cell. The nuclear membrane is a double-layered membrane with pores that monitors exchanges between the cytoplasm and the nucleus. The nucleolus is a round body that holds a large amount of RNA and it's attached to the nuclear membrane. Chromosomes are tiny rod-shaped bodies that are composed of DNA. In humans, there are 46 chromosomes, and of that, there's 23 pairs. With the cell membrane, it's a structure that surrounds the cell. It's a single cell membrane. It functions as a barricade by protecting cellular components from the environment, and it controls passage of water and other materials in and out of the cell. As far as cytoplasmic organelles are concerned, there are several that we're going to talk about. The first one is in the endoplasmic reticulum. This is a channel or series of channels, and its functions are that it allows the nucleus to communicate with the cytoplasm. It controls the passage of molecules such as RNA from the nucleus to the cytoplasm. It serves as a bridge, and it has the process of synthesis of lipids that occurs within it. Mitochondria are large bean-shaped structures. Their function is that they digest macromolecules to produce fuel for the cell, and they're considered powerhouses of the cell. Lysosomes are small pea-shaped sacs containing enzymes, and they're capable of digesting ingested material or damaged tissue and sometimes the cell itself. This is a digestive organ of the cell or the cellular garbage disposal. Ribosomes are small dot-like or spherical structures made up of protein and RNA. And the function for them is that they are the site of protein synthesis, so these are the protein factories for the cell. Cilia are thread-like projections that are through the cell membrane. Their function is to sweep materials across the cell surface. The flagellum is one thread-like projection through the cell, and its function is enables the cell to move. An example of a cell with a flagellum would be a sperm cell. In protein synthesis, DNA has the molecular code, which is determined by the sequence of the base pairs that identifies proteins the cell will make. A codon, which is a series of 3 base pairs, identifies one of the 22 amino acids available for protein synthesis. And the message is then passed in the nucleus from the DNA to the mRNA, which is the messenger RNA. The messenger RNA leaves the nucleus by way of the endoplasmic reticulum and goes to a ribosome, where the message is transferred to the tRNA. The tRNA goes into the cytoplasm to look for amino acids. The tRNA attaches to the amino acids and carries it to the ribosome, which join with other amino acids by peptide bonds to form the protein molecule. You may remember that we went over this chart when we were in class, that we talked about how you would start off with a nucleus, which had both the DNA and the mRNA. The DNA gave the message to the mRNA, and the mRNA went across the endoplasmic reticulum to get to the ribosome. Then it would give the message to the tRNA, which would go out into the cytoplasm to get the amino acids and carry them back to the ribosome to make the protein. So just wanted to remind you of that process that we talked about. We have 2 types of cells. There's somatic and genetic. Somatic cells are the cells of the body, such as nerve cells, not body cells, I meant to say nerve cells, blood cells, skin cells, any cell except for reproductive, and they undergo the process of mitosis. Genetic cells are reproductive cells, and for example, sperm cells and eggs, and they undergo the process of meiosis. The cell cycle is comprised of 4 processes, and that is, or stages, and that is the M, which is mitosis, G1, S, and G2. G1, S, and G2 are part of interphase. And then mitosis is where there's prophase, metaphase, anaphase, and telophase. Interphase is the period of cell growth during which a cell produces a second set of chromosomes and stores energy. During interphase, there's G1, S, and G2. G1 is referred to as the pre-DNA synthesis phase, during which RNA synthesis occurs. It lasts from just a few minutes to several hours. During the S phase, DNA synthesis occurs, and this is where chromosomes change from having 2 chromatids to having 4 chromatids attached to a centromere. This can last a maximum of 15 hours, which makes it very slow and very, very low sensitivity. G2 is the post-DNA synthesis phase, and it's the RNA and protein synthesis that occurs during this phase. It lasts from 1 to 5 hours. So here's the S phase. This is the pre-S phase on the left-hand side at letter A, where there are 2 chromatids attached to a centromere. On the right, we have 4 chromatids attached to the centromere. That's after the S phase. M, which is mitosis, is the division of somatic cells. We're going to have one cell with a diploid number, which is 46 chromosomes, that divide to form 2 cells with a diploid number, which is also 46. The daughter cells have the same genetic material as the parent cell. In mitosis, we have prophase, which is the first phase. The nucleus swells, the DNA becomes more prominent, the nuclear membrane disappears, the centrioles, which are contained within the chromosomes, move toward the opposite sides of the cell and organize the spindle fibers, which extend across the equator. Metaphase is where pairs of chromatids line up across the equator of the cell, and the centromere of each pair is attached to a spindle fiber. Now, during this phase, we're going to best analyze chromosome damage under a microscope. Everything's lined up across the equator. During anaphase, the centromeres split. The spindle fibers contract and pull the chromosomes one set toward each pole or spindle of the cell. They're moving away. And then telophase is where the chromosomes reach the poles of the cells and the DNA uncoils. The nuclear membrane reforms around each set of chromosomes, and the cytoplasm is divided into two separate parts. Meiosis is a division of reproductive cells. So this type of division is called reduction division because we're going to go from 46 chromosomes down to only 23, which is exactly half. So the two types of meiosis are spermatogenesis and oogenesis. In spermatogenesis, we're going to go from one cell with a diploid number chromosome, which is 46, to form 4 cells with a haploid number, which is 23. In oogenesis, we're going to have one cell with a diploid number, which is 46 again, divides to form one cell with the haploid number instead. And so here's the process of meiosis. In the spermatogenesis, we have one cell with the 46 chromosomes that forms 4 cells with a haploid number, which is 23 chromosomes. In the process of oogenesis, which is at the bottom, we're going to take one cell with 46 chromosomes, which is called an oogonium, and form one cell, which is called an ootid, at the very end with only 23 chromosomes. This concludes Part A on human biology.